I am Dr. Ila Shukla, and today's lecture would be covering the sources and the types of microbial contamination and their assessment. So, coming to the next slide, it says about it says about the contents that this slide all this lecture would be covering. First is microbial contamination, then the type of microbes, sources of contamination, types of microbial contamination, cross contamination, how it happens, principle of detection, how we can detect the contamination, then assessment procedures that are responsible for this kind of detection, then signature biolipids and the biochemical assessment methods, then assessment by immunoassays. Immunoassays are also used for their assessment, and then comes the biosensor assessment. So, coming to the first slide of this lecture, that is the microbial contamination. It is defined as the presence of microorganisms like bacteria, virus, fungi, algae, etc. in any food or pharmaceutical preparation. Any presence of microbe in food or pharmaceutical preparation is referred to as microbial contamination, because it is never like that microbes should be present in any kind of preparation that is being consumed by us. This poses a serious concern since metabolites synthesized by such contaminating microbes are often highly toxic. These microbes synthesize particular kind of metabolites for their own consumption, but these metabolites are toxic to us. Moreover, often they develop some kind of chemicals which are for their own defense system, which should not be used by us because these are also toxic to us. So, another point is contamination leads to change in taste, smell, color and efficiency of any given product. That particular preparation which is being consumed by us is, if it is microbial contamination is there, then its smell would change, its color would change, maybe it could become light color, dark colored, or turbidity happens, and efficiency will drop. Efficiency refers to the amount of effect that the particular substance or the drug would be having over that particular disease would not be there. Coming to the next slide. So, this slide says about the type of microbes. There are very type of microbes. We classify them according to their properties, according to their structure, according to their characteristics. So, first what the characteristic we have taken is there. First is the cellular and another is the acellular. Cellular refers to they are having particular cell organization, that particular cell membrane is there, nucleus is there and other organelles are there. And acellular refers to they are not having complete cellular setup, that is nucleus is absent, maybe the cell wall is absent, it is like that. So, when it comes to cellular microbes, first is fungi, example is yeast or mold, another example is protist, under this category comes algae, protozoa and slime molds, and then comes the bacteria, the example is Escherichia coli, it is very common example, then comes archaea, example is methanogens. So, these are the group of compounds with their examples which are the cellular organisms. Then comes to the acellular organisms. What happens in acellular organisms? The acellular organisms first category is viruses. Viruses, why? Because they are just having protein structure, the capsid and the nucleic acid, that is it could be RNA or DNA. So, these are not having complete cellular setup, that is why we call them acellular. Then comes to the viroids. Viroids are just the genetic material. When they are outside the living system, they are dead, they would not act, they would not perform anything, but when they enter the human being or any organism that is living, they start developing, the genetic material start replicating, they use the host machinery for their replication. So, another example is the this viroids is RNA. Then comes satellites. Satellites is a group of microorganisms, or we can say the acellular organisms which are having nucleic acid enclosed in the protein shell. Viruses are having proper capsule, protein capsule, they are having characteristic proteins over them and the nucleic acid is inside it. But when we talk about satellites, it is just like a small protein shell around the nucleic acid. Then comes prion. Prions are just the proteins, those are highly infective. If outside the body, they are behave like non-living, they are behave like ineffective, but when they are placed in the living system, they start replicating by using the host machinery. So, these are the type of microbes which may cause contamination. Moreover, these acellular organisms often do not cause any kind of changes in the odor or the smell when they are in the preparation. It is very difficult for us to find out whether the particular substance is being having microbial contamination or not, if these are contaminated by the acellular microbes. Why? Because these are active only in the host machinery or inside the living organism. So, it is very important for us to keep a pace over these microbes and keep a check over the microbial contamination. Coming to the next slide, it says about the sources of contamination. We have categorized here into the 6 categories, that is the facility, equipment, process, material, utilities and personal. Since source of contamination categorized here are only 6, but there could be more sources, because where we are preparing these pharmaceutical preparations, it could be like the since we enter the room for the preparation of the pharmaceutical product, we have to keep in mind that we are already carrying lots and lots of microbial load. If we do not check it there, it will enter into the place where the pharmaceutical product is being synthesized, and if it enters there, the microbial load present there would add into it, and then the product would be microbial contamination happen. So, it should not be there. So, while we are going for any kind of pharmaceutical preparation and we are going for particular procedure, for that purpose specially designed facilities and equipment should be used, proper protocol should be followed. So, first thing, the particular person which is synthesizing or which is involved in the production of pharmaceutical preparation, the person has to keep in mind that the particular environment he is travelling through is having loads and loads of microbes. So, when it enters that particular area where the pharmaceutical preparation of the production is going on, the person should go and first of all get sanitize itself. First of all, the clothes should not be same, that the clothes being used outside that area, the clothes should be the sterilized one, that the clothes which will be used in that particular area where the preparation is being done. Then comes facility. Facility, it is like air conditioning or anything else, the proper air conditions should be used, the filters should be cleaned properly. Then equipments, moreover the equipments are often become the reason behind contamination. Why? Because these are not cleaned after every use. So, these has to be cleaned after every use if we want the contamination to reduce. Then process, it is like if it is open, if you are preparing syrup in an open container, definitely it will be having more contamination compared to the closed one. Then comes the material, the raw material which was used was not properly sterilized before use, or the packaging was not that appropriate. Then comes utility, maybe the water and the gases that are being used in it are not properly not coming from a proper channel. So, that is why this contamination may happen. So, coming to the next slide. So, like source of contamination could be, as you can see in the image, if we shake hands, like it is like we sanitize our hands, we shake hands, but sanitization is not exactly what happens. Like if you shake hand, even after sanitization, there are chances of the contamination or there are chances of the transfer of microbial load, because sanitizer does not clear all the microbes from your hand. So, personals which are working in the pharmaceutical area need to check it that they should not shake hands or they should not touch door handles or something. When they enter the pharmaceutical area, those should be wearing properly sterilized gloves, and they should avoid touching any area until unless they reach that particular pharmaceutical that slab or something where the preparation is being done. And the person has to go through the air curtain so that the dust should be blown off before it enter that particular area. So, the source of contamination could be air carrying dust, outer skin of the personal. That is why you must have observed that the preparation area is where the preparation area is there, the personal is wearing complete from top to bottom, it is covered, no skin is visible. They wear goggles, they wear complete gloves, they wear PP kind of kits. Then person walking, example of how contamination may happen, a person's walking will liberate 5000 bacteria per minute. So, it is the range, it goes beyond the range. So, we have to keep a very strict check over it. Then sneezing should not be there, manufacturing process itself can generate contaminants. Moreover, if the particular machinery is not being cleaned properly, that substance which was spillage over there was there only, then that would also attract the microbes. Coming to the next slide. This slide is showing how the dust particle may carry different microbes, because the size of microbe is much lesser than the dust particle. You can see the dust particle is having particular size from 0.5 micrometer to 500 micrometer, and the virus size is 0.006 micrometer to 0.03 micrometer, which is really less. So, a particular dust particle may carry several viruses as well as several bacteria. So, that is why it is very important for us to keep a dust free environment where the pharmaceutical preparation is going on. Coming to the next slide, it says about the types of contamination. So, types of contamination could be direct contamination, like from the direct facilities or the substances or the machineries that are present over there, like it could be heating, ventilation and air conditioning systems. Whatever is the environment that is being maintained in that particular pharmaceutical preparation room, if it is not monitored time to time, like the air conditioners, those are not cleaned time to time, the filters are not changed properly, the dust keep on settling over it, and after a particular time the filter get expired and the dust may enter that particular area where the preparation is going on. So, it has to be checked again and again. Then comes the cross contamination. Cross contamination is how microbes can spread. Like we develop a particular sterile environment in a area where the pharmaceutical preparation is going on, but what happens, everything is good, HVAC system is very good, but a person coming from outside is having that microbial load and forgets to go through the air curtain. It has changed his clothes, it has worn the PP kit, but forget to go through the air curtain and carry that dust with itself. So, this is the cross contamination. That area is not having that contaminant, but the contaminant is coming from somewhere else. So, passing of microorganisms or other harmful substances indirectly from one sample to another through improper or unsterilized equipments. Moreover, I give you example of autoclave. Like autoclave is there, we do not clean it from outside, like we do not sterilize or we do not sanitize it properly, its lid and its container. We open that lid, that unsterilized and unsanitized lid, we open the lid, we keep our culture media into it to get it sterilized. What happens is that the media is sterilized, that is fine, but when we open it, we are touching that lid which was not sanitized. The microbes over that come onto our hand, with those hands only we pick that culture media, we take it out, we transfer to the another plate. What we have done? We have done cross contamination. The culture was not having that contaminant, that area was not having that contaminant, but because of the unsterilized equipment or unsanitized equipment, the microbial load was there and it was transferred to that particular culture. So, coming to the next slide. So, these are the type of contaminants, like it could be a contamination, it could be direct, like directly from the facilities over there, or it could be because of the improper management system, like the equipments are not properly sterilized or the handling techniques are not good. So, this is how direct contamination or cross contamination may happen. The contaminant from the environment operator and the contaminant from the equipment directly they are coming, that is direct contamination. The product of environment operator से कोई product निकला and that product is responsible for the contamination, that means cross contamination is happening. So, coming to the next slide, these are the images which are defining how cross contamination occur. You can see the personal, in the first image the person is walking with the shoes unclean and this dust will go into that particular area where the pharmaceutical preparations would be done. So, this dust then slowly what happened? Many people say that we have sanitized our hand. What is like if we do not clean our shoes, it is okay, na? We are not going to sit on the floor. No, it is not like that. The dust that has entered the room will keep on moving because of the air in that area. Air is continuously moving because we are moving. Or moreover, we switch on the fan or air conditioners, what happens is that air starts moving, that dust starts moving, and that dust slowly comes into the air, and when it comes into the air, slowly it will start contaminating the product that is being synthesized, and that contamination would go totally unnoticed because we didn't notice that our shoes were not clean, the shoes were carrying the dust. So, this is how cross contamination may occur. Moreover, in the second image you can see the spillage, like here all over this you can see that, in this you can see that the spillage is there, powder is spilled over it, and you can see that the person is doing the work in that area only. What will happen? Although the person is doing it in a aseptic area, but the powders from the different formulations may get mixed up, so it should not be there. This is also a reason for cross contamination. So, coming to the next slide, very clear images here, like if the equipments remain unclean, what will happen? The proper use was not there, the powder was taken out, particular formulation was being done. Maybe the powder was to be used for the tableting machine. The powder was taken out. What happened? The powder was spilled over the equipment, and what happened? The another lot will come, another lot will also be placed into it, and the machinery would start. The powder spilled over here was attracting different microbes from the environment. Maybe if the powder was not here and the area was completely sanitized, the microbes would not have come up to this equipment. But since powder was attracting the microbes, the another lot was capped into it, the another lot would also be carrying the contamination, and this contamination would completely go unnoticed because it was in the preparation phase. Later it will be developing microbial spoilage if the microbe starts surviving or proliferating in it. So, you can see the spillage over here. Moreover, the open system that I just talked about, if the system is open, if the preparation is open, the microbial load will be more. Here also the spillage is here. Here this equipment is not properly cleaned. You can see the marks of the particular chemical in it. So, coming to the next slide. So, how we can minimize the cross contamination? Cross contamination can be minimized by a very easy method. It is just like a basic awareness that the personnel should have. If a personnel is having a basic awareness about the contamination, the majority of the contamination can be avoided. Like first is personal procedures, that is skilled manpower and technical awareness. If technical awareness is there, such kind of things may not happen. The person would be like, if a particular skilled worker is there, if the technical awareness is there, every technical assistant or every technical worker will be cleaning all these things, then the next lot will be placed. Then coming to the next point, that is adequate premises, proper layout and area classification. When we talk about area classification, it is like the particular area is aseptic one, a particular area is having a particular concentration of the dust or that particular amount of the dust particles are allowed into it, and a particular area is there that no dust is allowed. These areas are classified according to the type of preparation being done. If a preparation which is not like we will be ingesting a preparation, it could be like we can manage to have some kind of dust or a limit of dust over there. When we are talking about the sterile products which will go directly into our blood, we need to keep a strong or a strict check over any kind of microbial contamination or dust. Dust, if I am saying dust, that means loads and loads of microbial contamination is there. Then the uses of closed production system. More the use of closed production system, more less will be the contamination. Why? If the particular preparation is being prepared in an open area, the microbes will directly settle over that surface. If the preparation is being done in a closed container, then the microbial load will be reduced by itself. Then adequate and validated cleaning procedures. Obviously, the workers which are doing cleaning over there, they should know what is the particular way that they should clean, because it is never like that just pour that cleaner and just wipe it out. No, there are particular ways that these should be wiped out, otherwise the microbial contamination or the load will be spread out throughout the whole area. Then appropriate level of protection of the product. Particular product, how it can be protected from different microbial contamination has to be maintained. Then correct air pressure cascade has to be developed. The facilities over there, that is air conditioning or heating, should be properly maintained so that no microbial load is coming from there. Coming to the next slide. So, here it says about the principle of detection, how we can detect the contamination. So, it could be recovery and concentration, purification and separation. By purifying the product and we separate out the contaminant, or by assay and characterization, by doing the assay and then finding out the particular characters whether present in it or not. Then there are the assessment procedures. First is signature, biolipid and the biochemical assessment methods. Then is the assessment by immunoassay, then assessment by biosensor. So, this slide says about the signature, biolipid and the biochemical assessment methods. What happened? Different kind of bacteria, different kind of fungi or different kind of microbes, I say, are having particular characteristic substances in their cell wall, or they metabolize, or they particularly prepare their substances, or they are having particular characters that will be responsible because of those substances. So, these kind of substances are characteristic and unique to that particular microbe, and if we detect them, if we are able to find out that whether the substance is present or not, we will be able to find out the particular kind of microbe or the bacteria or the fungi is present over that or not. So, contaminants contain distinctive macromolecules that aid in identification and detection. As I just told you that the contaminants, whatever the contaminants are there, are having particular kind of macromolecules. If we can find out what are those particular macromolecules, we can detect the contaminant. So, cell wall component assay for the lipopolysaccharides, muramic acid and the assays for signature biolipids. Signature biolipids specifically refer to the substances those are absolutely unique to a particular microorganism. So, we call them signature biolipids. That means that if biolipid A is present in a particular contaminant, that means the microbe present there could be X. It is like that. So, thin layer chromatography is used to separate and purify and then identify the microbial lipid. What we do? We take the contaminant, we try to concentrate the contaminant from the different preparations. When it get concentrated, we search out for the kind of unique biolipids or the signature biolipids found in these microorganisms. Then we go for TLC, that is thin layer chromatography. We prepare the plates, we run that sample over that plate and then we purify that particular spot that is coming on the plate and identify that whether the particular kind of microbial lipid is present over it or not. Then comes the assessment by immunoassay. Immunoassays are very common like ELISA and further many assays are there which are used to detect the particular kind of antibody or antigen present in that particular preparation or not. What happens is that we take that preparation, we find out for the particular kind of antigen or antibody in it, we place that accordingly, we place that complementary antibody or antigen into the substance. If the clogging is there, we run it on the plate. So, if the clogging is there, we read the plate and we find out whether the contamination was there or not. So, immunofluorescent detection by microscopy is a specific way to detect the microbes. Immunofluorescent microscopy refers to that the sample would be fluorescing, that sample would be showing fluorescence when we observe it. So, immunofluorescent refers to the we place the immunoglobulins and then the particular kind of contaminant would be showing fluorescence. So, we detect it as per the microscopy. Antibodies directed against antigens of a target pathogen and labeled with a fluorochrome by direct immunofluorescence. For direct immunofluorescence, what happens in it? The antibodies जो particular antigen के लिए responsible हैं उनको हम we label it with the fluorochrome. Fluorochrome is a substance that will be fluorescing, that will be producing fluorescence, that will be detected by the microscopy and that it will directly fluoresce. So, we will just place that plate under the microscope and we will observe it. These fluorescent antibodies reacted with the target microbe, these react with the target microbe, then the microbe preparation is washed to remove meted fluorescent antibody. What happened? The fluorescent antibodies react with these target microbes. The target microbes are the one which are causing contamination and then they react with it and get attached to it. Then we wash the plates, the excessive kind of dye and the excessive kind of these fluorescent antibodies are washed off and then the plate is ready for study. They examine for the target microbe by the ultraviolet light microscopy. Then we observe this plate under the UV light microscopy and find out if the contamination is there or not. If it is there, that what kind of microorganism is present in it. Then coming to the next slide, it is the image that is showing the immunoassay. It is the ELISA reaction. You can see this is the surface, and these are the captured antibody over the surface. We add the antigen into it, then the labeled antibody addition is done. After that, the labeled antibody get attached to it, the antigen that was added priorly, then the substrate is added to it. You can see the substrate causes this signal detection is, that means that it will be fluorescing, it will be producing fluorescence which will be detected by the light microscopy. Then comes assessment by the biosensor. This biosensor this assessment by biosensor refers to the electrochemical and the further biological or physicochemical properties of the microbes that they produce when they are placed in a particular kind of transducer. When they are placed at the particular kind of transducer, the microelectronics start working and then they find out the electrochemical properties like by the amperometry or the potentiometry. If the optical, we say they may produce fluorescence, they may produce bioluminescence, or they may produce some kind of colorimetric changes, or it could be like microbial fuel cell. So, these are the electrical signals which generate. when these microorganisms settle over the transducer, and then with the help of this microelectronics, what the characteristics we find out is that what kind of activity they are having, what kind of electrochemical reaction they are having, we detect by either amperometry or potentiometry. Then the optical changes could be like just the prior one, as the fluorescence was being done, the fluorescent, if it is fluorescent or not, if bioluminescent means that they are having their own some kind of substance which causes luminescence, which causes fluorescence. So, bioluminescence is there or not. Colorimetric changes, maybe the color changes are observed in it. So, these are the basic changes which are observed for the detection of the contamination by a particular microbe. So, with this I will end this lecture. Thank you.